Good day, students. I am Dr. Joseph Chua, and in this lecture, we'll be talking about blood physiology with a special focus on red blood cells. And these are our learning objectives for this lecture. We'll be talking about the components of blood. We'll be talking about the structure, characteristics of the erythrocyte. We'll be talking about how the red blood cell is formed, how it matures, also about hemoglobin, the oxygen dissociation curve, iron metabolism, RBC destruction and recycling, some diseases related to the red blood cells, and also we'll be talking about blood typing. So first, what is blood? What is blood? So according to Boron, blood is a complex fluid consisting of a plasma, which is an extracellular fluid rich in proteins, and formed elements, which include your red blood cells, white blood cells, and platelets. So blood is a fluid. It has a more fluid element called the plasma, and it also has cellular elements that we call the formed elements. Interestingly, blood is considered a connective tissue. It is considered a specialized connective tissue, and you'll study more about its classification in anatomy. And blood comprises 7 to 8% of the body weight in humans, a little bit less in females, and a little bit more in males. And you'll also see later that there's not only a difference in blood volume, but also a difference in red blood cell count between males and females. And this difference is usually attributed to testosterone, although there is still some debate as to whether testosterone is the primary reason for this difference. But for now, it's important to remember that there is this difference between the sexes. So if you look at this, 70 ml per kilo or 80 ml per kilo, and if we assume that a typical Filipino is around 50 kilograms, so the typical blood volume in a Filipino would be around 4 liters. So blood is composed of 55% plasma and 45% formed elements. So if you take a blood sample, put it in a test tube, and centrifuge that blood, you will see that the blood separates into these different layers. So the layer on top is your plasma, while the one on the lower layers, you will see the formed elements, the cellular elements of the blood. So most of these formed elements are actually your red blood cells, and the small sliver called your buffy coat, is a whitish layer that we call the buffy coat, is where you will find the white blood cells and the platelets. So this proportion, the proportion of red blood cells in the blood, is also known as your hematocrit. So the hematocrit is the fraction of the column of blood occupied by the red blood cells. So of course you compute this by looking at the height of the red blood cells divided by the total height of the column of blood. So your hematocrit is around 40% for females and 45% for males. Again, as we mentioned, males generally have a higher amount of red blood cells compared to females. So basically what we're saying is 40 to 45% of your blood is actually composed of your red blood cells. For newborns, this proportion would be a little bit higher at 55%. So let's look at plasma. So plasma is the pale white watery fluid portion of your blood. It's 91% water, 7% proteins, and 1% other components. So among these proteins, the major one is your albumin, followed by globulin, fibrinogen, and other regulatory proteins. So we will look at the functions of these proteins in the next slide. So 1% other components like your electrolytes, so sodium, potassium, chloride, etc. Nutrients such as your glucose or your amino acids, lipids, and wastes. And serum is differentiated from plasma. So when we say serum, that means plasma without clotting factors. Okay, so maybe you've already experienced, say, leaving maybe the medtex in the class. Maybe you've already experienced leaving a tube of blood, just leaving it alone and waiting for the blood to clot. So when the blood clots, you might have noticed that there's a portion that is more watery. So that portion that is left, that fluid portion that is left after the blood has clotted, that is your serum. That is plasma without clotting factors. So looking at the different proteins in the blood, so your albumin, so because it's the main protein component of the blood, it's the one that is the major contributor to the oncotic pressure of the blood. Again, recall when you discuss osmosis, when you discuss transport across the membranes, when we say oncotic pressure, that is osmotic pressure due to large proteins or proteins and other large molecules. Okay, so if a solution has a very high oncotic pressure, that means that the fluid or the water will tend to stay inside the solution and will not likely flow out or move out of the solution. Okay, so the oncotic pressure represents how well that solution is able to retain its water. And also your albumin also helps transport lipids and hormones and other drugs. Sometimes drugs can also bind to albumin. So this is an example, a picture of edema. So when the person has edema, so we see that this patient's foot is all puffy. It looks very puffy, and that's because the interstitial space has a lot of fluid. And one reason for edema is if there is a low amount of proteins, especially albumin, in the blood. If the blood does not have a lot of proteins, does not have a lot of albumin, so the oncotic pressure in that blood, it will become very low, and so the water, instead of staying in the blood, will now move into the interstitial spaces, thus causing this very puffy appearance. So the fluid is now leaking out of the blood and accumulating on the foot in this case. Okay, so that causes this appearance of edema. And the next protein in the blood is your globulin. It also has a transport function. It helps transport ions, hormones, lipids, etc. And also an immune function. Maybe you've heard of immunoglobulins, your antibodies. So those are examples of globulins. Fibrinogen is an important protein for blood clotting, and you will be introduced to fibrinogen and all of the other proteins and clotting factors in your lecture on hemostasis. And you also have these regulatory proteins such as enzymes, clotting factors, and hormones in the blood. And plasma proteins are made mostly in the liver. And what's the importance of this fact? So if a patient has a liver problem, that might affect the levels of proteins in the blood. So someone with a liver problem might not be producing enough albumin, so that might cause edema. Might not be producing enough clotting factors, so it could cause clotting problems if there is a liver problem. What are the functions of blood? We know, of course, that blood transports gases, nutrients, hormones, waste, and cellular products all throughout the body. It restricts fluid loss. Again, that's due to the oncotic pressure in your blood due to the protein components, especially albumin. It helps to regulate body temperature. So as blood flows throughout the body, it helps equalize the temperature throughout the body. It helps distribute warmth to the more distal portions of the body, right? Maybe you've experienced maybe you've noticed that if a part of your body it looks pale or is not receiving enough blood, then that part of the body starts to feel cold. It also is important in the regulation of pH and the ionic balance in your body. Okay, so you will study more about this when you look at the renal module and the acid-base lecture. Now let's look more specifically at red blood cells. So if you look at this picture, this is what you will see if you look at a blood smear under a microscope. So these are your red blood cells. The major function of your red blood cells is to transport hemoglobin. So each red blood cell contains a large amount of hemoglobin, and the importance of hemoglobin is that they are oxygen carriers. So hemoglobin carries oxygen, which is then transported by your red blood cells throughout the entire body. Other functions, they're very important also in carbon dioxide transport. We'll expound on this in a later slide, and you'll also study more about this when you go to the respiratory module. And it's also important in the acid-base balance of the body. So the red blood cells help in buffering the acids and the bases in the body. Part of this is due to hemoglobin, and part of this is due to its role in CO2. Again, as you will explore in a later lecture on the acid-base balance, the levels of CO2 in the body are very important for acid-base balance. So the average number of red blood cells is around 5 million per milliliter of blood in males, and around 4.7 million per ml of blood in females, so a little bit less in females. Again, this may be related to testosterone. Now let's focus on the structure of your red blood cell. So again, here is the red blood cell under a microscope, and here is an illustration of the red blood cell. So what's immediately apparent is that their shape is what we call a biconcave disc. So it's a disc, and the upper lower surface, or both surfaces, are concave, so it's a biconcave disc. And again, looking at this illustration, we see that the cell is very plain. The inside of the cell looks very plain. Unlike other eukaryotic cells, other cells, when you look at them under the microscope, you see some features inside the cell, maybe a darker area for the nucleus and the other organelles. But for the cell, it looks plain. The red blood cell, it looks plain because it does not have a nucleus nor other organelles. Diameter is 7.8 micrometers, 7.8 micrometers, and this size is actually just enough to squeeze through the smallest capillaries. So the very smallest or narrowest capillaries actually have a diameter that's a little bit smaller than 7.8 micrometers. However, as you will see later, your red blood cell is very flexible. They can deform, so they can it's possible for them to pass through those very narrow capillaries. Actually, in the narrowest capillaries, your red blood cells pass through them in single file. The thickness is 2.5 micrometers in the thickest area, so in the more outer portion, and in the center, of course, it's narrower because of the biconcave shape. And as I said, they're easily deformed. One reason why they're easily deformed is because of their biconcave shape. So this shape also allows them to bend, to deform without actually breaking their cell membrane or damaging the cell. And this biconcave shape also allows them to have a larger surface to volume ratio, so that that depression in the biconcave shape allows for a greater surface area. So surface area is important because, again, the main function of your red blood cells is to help in the diffusion or delivery of gases in the oxygen and carbon dioxide balance. So it allows for a greater diffusion area for those gases. So as we mentioned, the red blood cell does not have a nucleus nor organelles. Thus, it cannot synthesize its own protein, and it cannot perform oxidative metabolism. So its ability to repair itself is limited, and its lifespan is thus limited. So because it does not have mitochondria, its ATP is generated exclusively by glycolysis. And it contains 2,3-diphosphoglycerate or 2,3-DPG, and the function of this is that it acts on hemoglobin to reduce the affinity of hemoglobin to your oxygen. And this is important because you want to reduce the oxygen affinity of hemoglobin when you need to release more oxygen out of the blood, out of the red blood cells, and into the other tissues, in order to deliver that oxygen to the tissues that need the oxygen. So we will discuss this a little bit more when we talk about hemoglobin. And your RBC also contains more glutathione than most cells, which helps protect it against oxidant damage. Of course, it contains a lot of oxygen, and that might contribute to oxidative damage, so glutathione can help protect against that. And it also contains carbonic anhydrase and the chloride bicarbonate exchanger, and this is related to its role in the transport of carbon dioxide and also in helping maintain the acid-base balance. So your carbonic anhydrase, this is an enzyme that rapidly interconverts CO2 and bicarbonate. So CO2, as it travels in the blood, it doesn't actually travel as CO2 gas, as dissolved CO2 gas. It actually travels in the form of bicarbonate, and that is mainly due to carbonic anhydrase. So it quickly converts the CO2 gas into carbonic acid, which then dissociates into hydrogen ion and bicarbonate ion, and this is the main form of CO2 as it travels in the blood and also in the red blood cells. So your chloride bicarbonate exchanger helps exchange chloride and bicarbonate. So the bicarbonate produced inside the red blood cell goes out in exchange for chloride. So this helps ensure that more carbon dioxide and more bicarbonate gets produced inside your red blood cell. Now for this part, we will talk about the red blood cell synthesis. And first, let's explore exactly where the red blood cells are synthesized. When we were embryos, the location or the site of red blood cell synthesis was in the yolk sac. As the pregnancy progressed to the middle trimester, this shifted to mainly the liver, with a little bit being made in the spleen and in the lymph nodes. And as the pregnancy progresses to the last month of gestation and onwards, so from onwards meaning birth, and then as the person ages, the site of red blood cell synthesis is now in the bone marrow. And as we see in this graph, so here in this graph we see the different bones which are involved in red blood cell synthesis and the percentage of their cellularity as we age. So cellularity means how much stem cells are in those in the bone marrow, the stem cells that we need for red blood cell synthesis. And we see that it changes over the years. Early on in our life, so in infancy, around that time, toddler, so the most of the synthesis was actually in the tibia and the femur, and the tibia and the femur had really high, very high cellularity when we were infants, but this drops. This steadily drops until we reach adulthood. By the time we're adults, there's basically no red blood cell synthesis happening in the tibia and in the femur. So what is now more active when we are adults? So we see that the bones that are more active for red blood cell synthesis in adulthood would be the vertebra, sternum, and the rib, mainly in the vertebra and the sternum. However, we also see that it actually steadily drops. It steadily drops. The cellularity drops as we age. So in the elderly person, their bone marrows are actually less cellular, contain less stem cells compared to a younger person. However, this does not necessarily affect the amount of red blood cells being produced. So even in an older person, in an elderly person, their red blood cells, their bone marrows can still produce an appropriate amount of red blood cells as long as the person is healthy. So what are the steps of red blood cell synthesis? Here in this illustration, we actually see a summary of how all of the different blood cells are synthesized, from your red blood cells, erythrocytes, your white blood cells, the granulocytes, monocytes, lymphocytes, and your platelets. And we see from the illustration that they actually originate all from the same ancestor. They all come from your pluripotent hematopoietic stem cell. So of course, you'll find this in the bone marrow. When your pluripotent hematopoietic stem cell divides, some of its population actually remain as pluripotent stem cells, so that you always have pluripotent stem cells in your bone marrow. And of course, some of it will become more committed, or what we call the committed stem cells. And these committed stem cells are the ones that will actually form these specific blood cells. So this family of blood cells can be divided into two lineages, the myeloid lineage and the lymphoid lineage. Now, how do we identify which cells belong in which lineage? So just think, lymphoid lineage just includes your lymphocytes, the T and B lymphocytes, and all of the other cells, red blood cells, the other white blood cells, neutrophils, eosinophils, basophils, monocytes, and your platelets, they are all part of the myeloid lineage. So for this lecture, we'll be focusing more specifically on how your red blood cells are synthesized. So given that all of those blood cells come from the same origin, what causes their differentiation? What causes a pluripotent hematopoietic stem cell to differentiate into several different blood cells? So this is due to these hematopoietic cytokines. So these are examples of hematopoietic cytokines. So the presence of these signaling molecules will influence the differentiation of your pluripotent stem cell. So these are some common examples: GM-CSF, or granulocyte macrophage colony stimulating factor, GCSF, granulocyte colony stimulating factor, MCSF, interleukin 3 and 5, TPO, thrombopoietin, and EPO, erythropoietin. And this is an expanded illustration of the one we had before, where you have the pluripotent hematopoietic stem cell and how it can differentiate into the different blood cells. And we just see here the role of those hematopoietic cytokines in influencing the differentiation of that stem cell. So we see the GM-CSF, GCSF, we see the IL-3, we see that IL-3 can affect actually differentiation into red blood cell and megakaryocyte and also basophil. We see EPO, erythropoietin. So erythropoietin is the one that's important to us in this lecture. So because it's the one that will actually influence, it's a very strong factor for differentiation into red blood cells. And your TPO, or your thrombopoietin, which is important in the differentiation into platelets. So these other factors, these other cytokines, you will study them more when you talk about the platelets and when you talk about the white blood cells. But for now, we will focus mainly, again, as I said, on erythropoiesis and, of course, your erythropoietin. So focusing now on the synthesis specifically of red blood cells. So what happens after the committed stem cell now starts to differentiate into a red blood cell? So we start from a proerythroblast, which becomes a basophil erythroblast, a polychromatophil erythroblast, an orthochromatic erythroblast, a reticulocyte, and then erythrocyte. So the erythrocyte is your mature red blood cell. So what's important here actually is the trend. What trends do we see? We see that as your cell matures, as it develops into a mature red blood cell, we see that the trend is for an increased hemoglobin. We see that it initially starts out as being somewhat blue, and later on it appears more and more red, and that shows that there's an increasing concentration of hemoglobin as the cell matures. And also we see the trend is a decrease in size. It was very large when it was immature, and the mature red blood cell is now very small. And also we see less basophilic material, or the nucleus and the organelles. We see that the organelles inside the cell, they shrink until it's almost insignificant in the reticulocyte, and we no longer see it in the red blood cell. So let's look at this in a little bit more detail. So your proerythroblast, this actually comes from your CFUE, colony forming unit erythroid, which is one of the committed stem cells shown in the previous slides. So your proerythroblast actually divides multiple times to produce multiple erythrocytes. So it's not just like one proerythroblast maturing, so it actually divides into multiple proerythroblasts to produce more red blood cells. And your polychromatophil erythroblast, as we see in the illustration, this is when it starts to get pink or red, because this is where your hemoglobin first appears. So it's also indicated in the name, polychromatophil. So polychromatophil is now a mixture of colors, not just blue, basophil, not just blue, but now a mixture of colors. You're now becoming a little bit more red, polychromatophil. And then your reticulocyte. So your reticulocyte, as the image implies, still contains remnants of organelles. So it's not yet fully mature. However, it actually already exits the bone marrow at this stage. So your reticulocyte will exit the bone marrow and enter the capillary through a process that we call diapedesis. When we say diapedesis, this means that the cell is able to squeeze through the capillary membrane in order to enter the circulation. So that's diapedesis. It's able to squeeze through the junctions connecting your the cells that cover your capillaries, that form your capillary membrane. And then while in the circulation, your reticulocyte will mature into the actual fully mature erythrocytes in around 1 to 2 days. And it only comprises around 1% of circulating red blood cells. So if the number is higher or lower than 1%, that is actually a good indicator, good clinical indicator of bone marrow activity. If there's too much reticulocytes, that might mean that your bone marrow is very active, maybe because you need more blood cells, you need to produce more blood cells. Or if it's too low, then that might mean that your bone marrow is not very active. Maybe there's a problem, maybe there's a deficiency in nutrients, and it's preventing the bone marrow from making new blood cells. Okay, so this is a very good clinical indicator for bone marrow activity. So how is your synthesis regulated? What will tell your bone marrow to produce more or to produce less red blood cells? So remember, the major function of your red blood cell is to transport hemoglobin, which is important for transporting oxygen. Thus, the most essential regulator for RBC production is tissue oxygenation. So the production of RBCs is closely related, very closely coupled to the level of tissue oxygenation. So how does that happen? To understand how the red blood cell synthesis is stimulated, let's look at this illustration. So we said that the oxygen level, the tissue oxygenation, is the key, or is an important factor that will help stimulate the production of red blood cells. So given these factors, examples of factors that decrease oxygenation, so you have low blood volume, you have anemia or a lack of red blood cells, low hemoglobin, poor blood flow, problems with your lungs. So all of these can decrease tissue oxygenation. And this tissue or oxygenation, when it affects the kidney, so when there is kidney hypoxia, this will induce the production of the hypoxia inducible factor 1. And if there's an increase in HIF1, increase in hypoxia inducible factor 1, this will actually stimulate production of erythropoietin in the kidneys. And we showed earlier that EPO, or erythropoietin, is one of the important differentiation factors that causes your stem cells to become erythrocytes. So again, a low oxygenation level in the tissues, or hypoxia, especially if it affects the kidneys. So this will increase hypoxia inducible factor 1 and increase erythropoietin production in the kidneys, and thus stimulating more differentiation of stem cells into erythroblasts, proerythroblasts, and then mature red blood cells. So other signals that can help stimulate the production of red blood cells would be your norepinephrine, epinephrine, and the prostaglandins. So production of erythropoietin is 90% in the kidney and 10% in the liver. That's why the kidney is very important in this flowchart, because it's the one that produces most of the erythropoietin. And that's why also if a patient has kidney problems, has kidney damage, that would also affect the levels of red blood cells in that patient. So someone with kidney damage would most likely also have anemia, especially if the kidney damage is severe. So once you stimulate the kidney to produce erythropoietin, the maximum production is reached within 24 hours and new RBCs will appear after 5 days. And EPO increases both the quantity and the speed of production, so it really has an effect, a large effect on the production of your red blood cells. And it's very important such that if you do not have any EPO, there's almost no RBC production. That's why we mentioned before that if someone has a severe kidney disease, that could lead to severe anemia as well. And if you have an excess amount of EPO, that could increase the normal rate of production up to more than 10 times the normal rate. So some clinical applications of this is if you have a patient with severe kidney damage, most of the time they're also given erythropoietin in order to stimulate the production of red blood cells and to prevent or correct the anemia. And in athletic circles, it is not allowed for athletes to inject themselves with erythropoietin. That is a form of doping. So some unscrupulous athletes would inject themselves with erythropoietin in order to hopefully increase their red blood cells and hopefully increase their performance in the sport. But again, as I mentioned, that is not allowed. I believe in most sports that is not allowed. So your EPO will stimulate the differentiation of your stem cell into an erythrocyte. However, of course, you need to ensure that the proerythroblast will properly mature into an actual erythrocyte. And there are two very important nutrients needed for that to happen, your vitamin B12 and your folic acid. So vitamin B12 and folic acid, these are vitamins that are very important in your DNA synthesis. Vitamin B12, this is a vitamin absorbed in the gastrointestinal tract, and very important for its absorption is what we call the intrinsic factor. So the intrinsic factor is a protein produced by your stomach which facilitates the absorption of vitamin B12. And your folic acid is closely related to your vitamin B12 because it requires vitamin B12 for synthesis. So if you lack one or the other, or maybe both, one symptom that will be seen in your RBC is what we call megaloblastic anemia. So here is a patient with megaloblastic anemia, and as we observe from the patient's red blood cells, there are red blood cells that are abnormally large, and the sizes of the red blood cells are also very unequal. You have normal looking red blood cells along with irregularly shaped or very large red blood cells. So these very large red blood cells and irregularly shaped red blood cells are signs that the RBC did not mature properly. And again, this may be related to deficiency of either vitamin B12 and or folic acid. Time for our first concept checkpoint. Take some time to review the following concepts, answer the following questions, and pause the playback before proceeding to the next slide. So just a clinical application. In one of the cases in the concept checkpoint, pallor was mentioned. So this is how we check for pallor or paleness clinically. So you can compare the patient's hand with your hand. So if the patient's hand is too pale, so that might be a sign of pallor. And you also check the palpebral conjunctiva. This one should be pink, but here in this picture it's very pale. So you can look at your own palpebral conjunctiva in a mirror. We are assuming that you have a normal RBC count. So the pinkness that you see, that is normal, and you compare it here with this picture, it's very pale. So these are two ways in the clinics to check for pallor. Now let's talk about hemoglobin. So we mentioned that this is the main component of your red blood cell, your hemoglobin. Your hemoglobin is actually a complex composed of 4 subunits, 1, 2, 3, and 4. And each of these subunits is composed of a heme plus a polypeptide chain. So here in this simplified illustration, this blue disc represents heme, and this squiggly line represents the polypeptide chain. Okay, so this is one subunit, heme polypeptide chain, one subunit, heme polypeptide chain, another subunit, heme polypeptide chain, and another subunit, heme polypeptide chain. What do we mean by heme? Heme is an iron-containing porphyrin derivative, and we'll look at the structure of this porphyrin in the next slide. So here is where you will find the iron, right? We know, maybe even since grade school, that hemoglobin contains iron. So where do we find that iron? We find that iron in your heme. It's integrated into the structure of the heme. So each complete hemoglobin molecule actually contains 4 iron atoms embedded into the heme. So polypeptides, this is also known as your globin portion, right? That's why it's called hemoglobin. So we actually can have 4 kinds of chains in your hemoglobin. We can have alpha, beta, gamma, and delta. And your hemoglobin A, which is what you will find in adults, comprises 95 to 98% of adult hemoglobin, is composed of 2 alpha chains and 2 beta chains. So this is the normal composition of your hemoglobin. The type of hemoglobin that's predominant actually changes depending on your age. So before you were born, when you were still a fetus, the predominant hemoglobin was hemoglobin F, which is composed of 2 alpha and 2 gamma chains. And then when you were born, and at around 3 months, the predominant hemoglobin now changes to hemoglobin A, your 2 alpha and 2 beta chains. However, there might still be some of the other forms of hemoglobin, because you still have some gamma chains and a little bit of delta chain. So you can have other types of hemoglobin, but the predominant one is your hemoglobin A. Now this picture just illustrates what happens in the heme group. What happens in the heme group when you have oxygen. So this is your heme. So this is the porphyrin structure, and this is your iron in the center of that structure. And we see that your heme is connected to your polypeptide chain. So this here is a deoxygenated hemoglobin. You do not see any oxygen attached to your ferrous, to your iron. But if you add oxygen, we see that oxygen is now loosely bound to your iron in the heme, and this is what we call oxyhemoglobin. So again, I'd like to emphasize that iron is just loosely bound to oxygen, is just loosely bound to iron, because of course hemoglobin needs to be ready to give up that oxygen in order to supply the tissues with oxygen. So how is your heme formed? I do not need you to memorize the steps in the production of heme, but I'd like to show you some of these chemical structures so that you have some idea of how this ring structure could have been formed. It started from glycine and succinyl-CoA, and you form this porphobilinogen, and they assemble into a ring structure, and then you add ferrous, the ferrous ion in the middle, and then you have heme. And again, as a review, your hemoglobin synthesis starts mainly here at the level of the polychromatophil erythroblast and proceeds until you have the mature erythrocyte. So let's look now at the function of hemoglobin. So recall the structure of your hemoglobin. You have 4 heme groups, and each with their associated polypeptide chain. And each of these heme groups can bind only 1 molecule of oxygen. and it can only bind oxygen if the ion in this heme group is in the ferrous or Fe2 plus state. And the chain, the polypeptide chain, whether alpha or beta, helps determine or affects the affinity of your hemoglobin for oxygen. So that's why in the fetal stage you have a different type of hemoglobin. You have the fetal hemoglobin because the fetal environment is very different from the environment of the blood when you're born and as you grow. So you need a different type of hemoglobin with a different affinity for oxygen in the fetal stage. Oxygen is bound loosely and reversibly. So we mentioned this before. It's bound to your ferrous, but it is bound via a coordination bond, what we call a coordination bond, a looser type of bond, and not an ionic bond. So the ionic bond, if you recall from your chemistry, is the one that's more permanent, but the coordination bond is much more loose and reversible. So oxygen, when it binds to hemoglobin, you call that oxyhemoglobin. But hemoglobin binds not only to oxygen, it can also bind other molecules. If it binds to carbon dioxide, you call it the carbaminohemoglobin. If it binds to carbon monoxide, it's carboxyhemoglobin, and this is we do not want this. This is an abnormal situation if most of your hemoglobin is bound to carbon monoxide. So this is part of carbon monoxide toxicity. And if the ferrous is transformed into ferric, so this is also bad for your hemoglobin. We call this methemoglobin. It's methemoglobin if the ferrous in the heme is transformed into the ferric state. So when it's in the ferric state, it cannot bind oxygen. So this is also a symptom of some forms of poisoning. You have methemoglobinemia. Let's explore a little bit more about how oxygen binds to your hemoglobin. When oxygen binds to hemoglobin, we observe this phenomenon that we call cooperativity, which basically says that as more oxygen binds to hemoglobin, the conformation or the shape of hemoglobin becomes more relaxed and it gains a higher affinity for oxygen. So what do we mean by this? So remember that each hemoglobin molecule can bind up to 4 oxygen molecules because you have 4 heme groups. So as soon as the first oxygen binds to that ferrous in the heme, the shape of the heme and of the polypeptide chain actually changes. So here we see oxygen binding to Fe2 plus, and we see that the shape of the heme ring changes from a dome to a planar conformation. Okay, so initially it was more of a dome shape, and then it transforms to more of a planar shape. So this causes also a change in the conformation of the polypeptide chain. So what is the effect of all of these changes in shape? So it basically somewhat relaxes. It relaxes your hemoglobin. It relaxes your hemoglobin, and this relaxation actually makes oxygen more likely to bind to hemoglobin. So as soon as the first oxygen binds to hemoglobin, the hemoglobin now becomes more attractive or more suitable for oxygen binding, and so this facilitates further oxygen binding until you fill up all those 4 slots in the hemoglobin. So this is what we call your cooperativity. The binding of oxygen relaxes the shape of your hemoglobin and facilitates more oxygen binding. You gain a higher affinity for oxygen. Now, building on that concept of cooperativity, let's look at the relationship between hemoglobin and your oxygen partial pressure. So this is your hemoglobin oxygen saturation curve. On the x-axis, we have the oxygen partial pressure, or the amount of oxygen in the blood, and on the y-axis, we have the percent O2 saturation of your hemoglobin, or how much your hemoglobin is saturated with oxygen. And we see that it has a sigmoidal shape, sigmoidal curve, a very steep rise at the start followed by a plateau. So the very steep rise is due to your cooperativity. Remember, in cooperativity, you have some sort of a positive feedback effect happening, right? Oxygen binds to hemoglobin, which increases its affinity for oxygen, which causes more oxygen to bind, which further increases affinity for oxygen. So it accounts for this very steep rise. And of course, you will plateau because each hemoglobin molecule can only accommodate 4 oxygen molecules, right? 1 oxygen molecule for every heme group, for every ferrous atom. So you will reach a saturation point. So what are some of the clinical implications of this sigmoidal shape? So this helps us know that humans can survive in different atmospheric oxygen levels. So whether you live in an elevated area or you live closer to sea level, your hemoglobin is almost always at the ideal saturation, at 95% and above saturated, whether you live in elevated areas or at sea level. Okay, so this is due to the plateau that can happen at a wider range of oxygen partial pressures. So the normal oxygen partial pressure is from 80 to 100. That's in the arteries. While in the veins, the oxygen partial pressure in the veins is at around 40, which would correspond to around 70% O2 saturation. Another implication is that the hemoglobin saturation can drastically drop if your oxygen levels are too low. So a practical application of this would be in our COVID times now, right? If you've ever taken care of someone with COVID, you know that you need to monitor their oxygen saturation, the hemoglobin saturation, right? We want the oxygen saturation to be above 95%. If it gets below 95%, 93%, 90%, 88%, we do not want that. We do not want the oxygen saturation to be consistently below 95%, especially if it's going closer to 90 or 88%, because at those points, we are now going to the more steeper part of the curve. And so we might experience a more drastic drop in your hemoglobin saturation if the oxygen in the blood continues to decrease. So besides your cooperativity, there are some other factors that affect the affinity of hemoglobin for oxygen. And the main principle here is that when tissues are metabolically active, certain factors will induce hemoglobin to let go of more oxygen. So the affinity of hemoglobin for oxygen is decreased given certain factors. And thus, it lets go of more oxygen, hemoglobin saturation decreases, and more oxygen is delivered to the tissues that need it. So if the tissues are metabolically active, what we see is what we call a shift to the right. So let's look at this illustration. So your green line, this is the oxygen hemoglobin dissociation curve, and the red line is the shift to the right. This is the oxygen hemoglobin dissociation curve if the tissues are more metabolically active. So what's the effect of this shift to the right? So given, say, the same PO2, for example 50, so at 50 PO2 in the usual hemoglobin oxygen dissociation curve, your oxygen saturation is at a little bit higher than 80%. But if you have a shift to the right, if the tissues are more metabolically active, what we observe is at 50, at the same PO2, the oxyhemoglobin saturation is now somewhat a little bit higher than 70%, around 75%. So this shows that your hemoglobin released more oxygen into the tissues. The oxygen affinity decreased. Now, what are these factors that will cause a shift to the right? If there is an increase in CO2, a decrease in pH, an increase in 2,3-DPG, and an increase in temperature. Now, CO2, pH, and temperature are all easy to understand. They are all related to your metabolism, right? If you use your mitochondria, if you perform glycolysis, if you utilize the Krebs cycle, you will be releasing CO2. And you will also be releasing acidic byproducts as part of your cellular functions. And also, you will see in a later lecture that CO2 and acidity is directly related. And temperature, of course, is an indicator of metabolic activity. You are producing heat. So all of these indicators of metabolic activity can actually cause the bond or the affinity between hemoglobin and oxygen to weaken. So it releases more oxygen. Now, what about 2,3-DPG? 2,3-DPG is a byproduct of glycolysis in your red blood cells. And for 2,3-DPG, more 2,3-DPG is produced if there is lower PO2. So if there's lower PO2, for example, the blood passes through tissues that have a low PO2, so the tissues need oxygen, more 2,3-DPG is produced, and 2,3-DPG actually stabilizes the tensed state of your hemoglobin. So it's like it helps push away oxygen from hemoglobin and stabilizes the tensed state of hemoglobin to further decrease the affinity for oxygen. So in this way, it helps release more oxygen into the tissues that need more oxygen. So of course, if the opposite is true, if you have low CO2, more alkaline, so less acidity, less 2,3-DPG, and a lower temperature, so these factors will cause now a shift to the left. So in a shift to the left, the hemoglobin has greater affinity for oxygen and less oxygen is released. Of course, the shift to the left will happen if the tissues are not very metabolically active. Let's focus a little bit on the relationship between CO2 and hemoglobin. So this is described by two phenomena, the Haldane effect and the Bohr effect. So what is the Haldane effect? The Haldane effect basically describes the relationship between CO2, or sorry, the affinity of hemoglobin for CO2, the affinity of hemoglobin for CO2. So in the Haldane effect, the observation here is that if you have an area with high O2 concentration, this will displace CO2 from your hemoglobin. So if you have carbaminohemoglobin and it goes to an area with high oxygen, the oxygen will displace that carbon dioxide. So carbon dioxide will be released and the carbaminohemoglobin now becomes oxyhemoglobin. So this is what you observe in the lungs. So when blood passes through the lungs, where you have a high oxygen, oxygen will displace CO2 and oxygen will then attach to hemoglobin. So you now oxygenate your hemoglobin and release your CO2 so that it can be exhaled. And when your blood now goes from the lungs and to the other parts of the body that now have a lower oxygen concentration, so this lower oxygen concentration actually increases the affinity of hemoglobin for your carbon dioxide. So as the blood passes through areas with lower oxygen, with lower oxygenation, the hemoglobin will then tend to pick up more CO2 from those metabolically active tissues so that it can be delivered to the lungs so that you can exhale your CO2. And then the Bohr effect describes the relationship of hemoglobin affinity for oxygen and the levels of CO2 and acidity. So as we described when we talked about the shift to the right and shift to the left, so if there is an increase in CO2 and an increase in your acidity or a decrease in pH, the hemoglobin affinity for oxygen will decrease, and you call that the Bohr effect. Now, what happens to the red blood cell at the end of its lifespan? Remember that your red blood cell does not have a nucleus and does not have organelles, thus it has a very limited ability to repair any damage that it receives during its lifespan. So its lifespan is limited to around 120 days, and as your red blood cell ages, the fragility increases. It becomes much more fragile. Remember that your red blood cells need to squeeze into very small capillaries as part of its normal function, and as it ages, it becomes now less able to do that. And so the final test of its fragility is when it passes through the spleen during the normal circulation. So when your blood passes through the spleen, the red blood cells are forced to squeeze into these narrow gaps in the spleen. Okay, so here we see a picture of red blood cells squeezing through these narrow gaps, these narrow spaces in your spleen. Okay, so the structure of the spleen is actually sort of like a net. So this net basically catches those red blood cells that are not flexible enough to pass through those gaps. So those blood cells that fail to pass through these gaps, or that break or are destroyed as they try to pass through these gaps, these are then phagocytized by the white blood cells in the spleen. So the hemoglobin contained in those red blood cells are also phagocytized and are broken down into the elemental iron and the porphyrin ring. And of course, the polypeptide chain will be broken down into peptides. So the iron will be recycled, and we will see that in the next slide. And the porphyrin ring will be converted to bilirubin, which will then be excreted in the urine and in the feces. Knowing that iron is very important for the blood, now let's look at how the body processes iron, the fate of iron in the body. So looking at these percentages, we see that 65% of the iron in the body is found in your hemoglobin. So of course, if you lose blood, if you have blood loss, of course you will be losing iron from that compartment. So in those who have menses, they will be losing 0.7 milligrams of iron daily. So when your hemoglobin is degraded, so the red blood cell, if the red blood cell is old and hemoglobin is degraded, we mentioned that this hemoglobin, the porphyrin ring, will be converted to bilirubin and excreted, and the free iron will be recycled. Now, what happens to this free iron? So for free iron, 15 to 30% of it is stored in ferritin. So ferritin is a protein that's able to store a large amount of iron. So this is the storage form of iron, your ferritin. If there's any excess iron that cannot be stored by ferritin, for example, if this is already saturated, if there's too much iron, it can be deposited in insoluble particles that we call hemosiderin. So 4% of iron is found in myoglobin. So this is the form of kind of like hemoglobin, but for your muscles. So of course, it has a similar function to hemoglobin, but specific for muscles. So of course, it also requires iron. Around 1% is found in various heme compounds. And your free iron, so from the storage iron to the free iron, we see that there is an arrow going to and from. So that you have interchange between that. You have iron being released from your storage. And when iron is released from your storage and it needs to travel to other parts of the body, it needs to travel through the blood, it will be in the form of transferrin. So transferrin is now the form of iron as it is in the blood. So it's the transport form of iron, your transferrin. So around 0.1% is found in the plasma, and so that transferrin can be endocytosed into your red blood cells to form to provide the iron needed for hemoglobin. And of course, if you absorb iron from your diet, you eat iron-rich food, that iron will of course first travel as transferrin before either being transported to your red blood cells or transported into storage. And of course, from here, from the plasma, you will also be excreting some amount of iron daily. To maintain adequate levels of iron in the body, the recommended intake per day for Filipinos is 12 milligrams per day for males, and for females with menses, 28 milligrams per day with an additional 10 milligrams if pregnant. And for females who don't have menses or who have experienced menopause, 10 milligrams per day. So why do we need 12, 28, 10 milligrams if the iron that we lose every day is only just around 0.6 to 1.3, 1.4 milligrams? And this is because the iron in our diet is only around 15% absorbed. So if you eat 12 milligrams, only around 1 to 2 milligrams is actually absorbed by the body, much less if you are eating a purely vegetarian diet. And this is because the iron in meat is much more readily absorbed than the iron in vegetables or grains. So do we need iron supplementation? For males, if you are already eating a balanced diet with a large variety of meat and vegetables and grains and legumes, you don't really need additional iron supplementation. However, consider supplementation for females with menses, especially if the menses are heavy or there's a history of anemia. And supplementation is much more essential for those who are pregnant, given the very high iron requirement in those who are pregnant. Now let's look at some clinical applications. So when you order a blood cell count for your patient, these are some of the values and parameters that you will see. Of course, you have the red cell count, or the amount of red blood cells per unit of volume. Usually expect a value of around 4 to 5, but this could be highly variable depending on the population and also depending on the specific laboratory. The hematocrit is also around 40 to 45%. Hemoglobin is the amount of hemoglobin in the blood per unit of volume of blood, around 14 to 15. So for males, again, it's a little bit higher. And the red cell volume, the mean red cell volume, or the MCV, basically tells you how large the cells are. The mean red cell hemoglobin basically tells you how much hemoglobin is inside every red blood cell, the average amount of hemoglobin in every red blood cell. And the mean cell hemoglobin concentration, or MCHC, which tells you about the concentration of hemoglobin in each red blood cell. So what's the difference between MCH and MCHC? So again, MCH is the amount of hemoglobin inside every cell, the average amount of hemoglobin inside every cell, but MCHC is the concentration. So it also takes into account the volume of the red blood cell, the volume of the red blood cell. So maybe the cell is very large, or maybe the cell is very small. So of course that would affect how much hemoglobin you expect to be inside the cell. So you can say that the MCHC, I think it's more useful because it not only takes into account the actual amount of hemoglobin, but also the concentration of hemoglobin. So it takes into account the volume of the red blood cell, which is also very important. And then your RDW, the red cell distribution width, tells you how similar are the sizes of the red blood cells. Typical value is around 13%. If this one is higher, it tells you that there is more variety in the sizes of your red blood cells. So maybe you have red blood cells that are too large, or red blood cells that are too small, and that maybe they form a significant amount of the population of the red blood cells. So that could cause a very high RDW. And then of course your reticulocyte count. Again, reticulocytes are the immature red blood cells, but they're already in the circulation. They've exited the bone marrow, and they're a good sign of bone marrow activity. If it's too high, maybe the bone marrow is too active. They're too low, maybe the bone marrow is not very active. So if the MCV is too high, we call that macrocytic, so the cell is too large. If it's too low, we call it microcytic, and then normocytic if normal. For MCHC, if it's too high, there's too much hemoglobin in the cell, it's hyperchromic, so the color is too intense, hyperchromic. But this is rare. Hyperchromic is rare. Usually we just see hypochromic. So hypochromic, there's too little hemoglobin, so hypochromic. But if it's normal, normochromic. For RDW, if it's too high, we call that anisocytosis. So the sizes of the cells are not the same. Basically, this is what the word means, anisocytosis. Now let's look at some illustrations of some of the abnormalities we mentioned in the previous slide. So I don't need you to memorize all of this, but again, this is just to illustrate, to give you a clearer picture of what we say when the red blood cells are abnormal. So this is your normal cell, normocytic, normochromic. So one abnormality can be the size. So you can have anisocytosis, like we mentioned earlier. The RDW is high. The cells are of varying sizes, or it can just be macrocytic or microcytic. You can also have abnormally shaped red blood cells, as seen in these pictures. You can have changes in color in your red blood cells, for example, hypochromic. You can have also the presence of immature cells, and so if you have too much reticulocytes, or maybe sometimes even the really immature red blood cells might be seen in the circulation. And then some other abnormalities, so some more unusually shaped cells, special forms of cells, or sometimes you can see abnormalities inside the cell itself. Now let's focus a little bit on some examples of red blood cell count abnormalities. So if you have a low count of red blood cells, you have anemia. You have a very high count of red blood cells, you have polycythemia. So first, let's look at anemia. What can cause anemia? So you can have anemia due to blood loss. Okay, anemia due to blood loss. Of course, if you lose blood, you will lose red blood cells. So in the acute phase, if you lose blood acutely, the body is only able to replace the plasma. Say someone got stabbed and lost a lot of blood. So in the first few hours, minutes, hours, of course, the body cannot yet replace the red blood cells. So how will the body adjust to that blood loss? It will just replace the plasma, just more replace it with more fluid. Okay, so therefore, the blood will be mostly fluid. The plasma proportion of the blood will be now much higher than your normal blood. So you have a lower RBC concentration. But for chronic blood loss, say so a person has cancer and is bleeding, has been having a little bit of bleeding now for several months or several years. So because of that constant blood loss, the body cannot absorb iron fast enough, cannot really replace the iron that's being lost because of the blood loss. And so what you have are small RBCs with little hemoglobin, or the microcytic and hypochromic anemia. While for acute blood loss, the red blood cells would still appear normal, because these are still your normal red blood cells, but there's now just much less of them because you lost some through blood loss. But for chronic, there's now a problem with the iron supply, and so the synthesized red blood cells look abnormal. They're smaller and they're paler. And then when we say hemolytic anemia, this is anemia due to destruction of your blood cells. So it might be because of fragile cells, so they rupture easily. For example, there's what we call hereditary spherocytosis, where there's a problem with the cell membrane of your red blood cells. So instead of being biconcave, they appear spherical, so they're much less compressible, so they are destroyed more easily. Or you can also have sickle cell anemia, where there's a problem with the protein chain of your hemoglobin. So it also affects the shape of your red blood cell. They assume a more sickle shape. So this could also be more fragile, or could also cause some clumping or obstruction of blood vessels. So that's hemolytic anemia. The cells are more fragile and are destroyed. So you can also have anemia due to disordered production. An example is aplastic anemia. In aplastic anemia, your bone marrow becomes mostly acellular. You no longer have the hematopoietic stem cells in your bone marrow. So this is normal bone marrow. See, it's very cellular. You see lots of dots there. So those are the hematopoietic stem cells. And this is the bone marrow of someone with aplastic anemia. You lose many or most or even all of the hematopoietic stem cells. So the bone marrow can no longer really produce a lot of blood. And so you have a lack of almost all of the different types of blood cells. So that's aplastic anemia, one of the more severe diseases that cause anemia. And of course, also megaloblastic anemia, we mentioned before, due to deficiency in vitamin B12 or folic acid. So you have improperly maturing red blood cells, so they are oversized, bizarrely shaped, and much more fragile. So this is anemia due to a problem with production of red blood cells. Now, what about polycythemia? So polycythemia, again, you have too much red blood cells. One form of polycythemia is polycythemia vera. Vera meaning true. It's true polycythemia, meaning you are truly overproducing red blood cells. So this is because of abnormal precursors, abnormal blast cells. And so they do not stop producing red blood cells. They do not respond properly to the cellular signals. So it doesn't stop producing not only red blood cells, but also the other blood cells, because again, they all come from the same precursor. So you have increased hematocrit, increased blood volume, increased viscosity of blood. So even the appearance of the patient would change because it really is engorged with blood. There's too much blood in the body. So there's low EPO because you are overproducing. Of course, the response, homeostatic response, is you produce less EPO. However, since the blast cells are abnormal, they don't respond to the signals, they ignore this. They don't really need EPO in order to divide and to differentiate into the blood cells. And you can also have secondary polycythemia. Secondary polycythemia is now more physiologic. This is polycythemia. You are producing more red blood cells because of a physiologic need. It's a physiologic adaptation to hypoxia. For example, if you live in very high altitudes, so again, in high altitudes, the amount of oxygen in the air is much lower. So perhaps to adapt to that environment, the body is forced to produce more red blood cells. Or maybe you have cardiac failure. The heart can no longer pump blood very well. So one way to adapt to that is to produce more red blood cells. So it's physiologic. You have hypoxia, so you have high EPO, therefore you are producing more red blood cells. So that's your secondary polycythemia. Time for another concept checkpoint. Try to answer these questions before proceeding to the next slide. Please pause the playback as you try to answer these questions. Now let's explore another clinically relevant topic, your blood types. So what's the basis for the different blood types? So the RBC cell membrane has at least 30 common antigens and hundreds of rare antigens. So that mix of antigens on the membrane of the RBC, that is what determines your blood type. So there are actually several different ways, several different blood type systems, okay? Several different ways to classify the RBC based on blood type. But the most clinically relevant blood types or the blood groups would be the ABO system and the RH system. So the ABO system, this is where you classify the blood into type A, type B, type O, and type AB. And the RH system is where you classify the blood into RH positive and RH negative. So when you have your blood type tested and you receive a result of A positive or say O positive, so that indicates your classification under the ABO system and the RH system. So first, let's focus on the ABO system. So your ABO system, as we said, has the blood types O, A, B, and AB. So these blood types, these represent what is on the cell membrane, what antigens are on the cell membrane. Antigens, or also known as agglutinogens, what is on the cell membrane. So if your blood type is O, there are no antigens on the cell membrane, at least from the perspective of the ABO system. If your blood type is A, you have the A antigen. If your blood type is B, you have the B antigen. And if your blood type is AB, you have both the A and the B antigens. So these blood types and these antigens, so this is the phenotype. Okay? This is what is exhibited by the cell. That's actually exhibited by the cell, the phenotype. And the genotypes are what's actually in the genes of the person. So looking at the alleles that code for these blood types, so we can label the alleles as O, A, or B. So if your blood type is O, your genotype is OO. You have both of the O allele. If your genotype is A, if your phenotype is A, your genotype, your genes is either OA or AA. For blood type B, OB or BB. And for blood type AB, you have both the A and the B allele. So this is an example of a co-dominant genotype. Both alleles are fully expressed, so you have both antigens. So if you have these antigens on the cell membrane, you also have corresponding antibodies or agglutinins in the plasma. So for type O, it does not have any antigens on the cell membrane, but it has both anti-A and anti-B. For blood type A, of course, it will not have anti-A, otherwise the antibodies will attack its own cells. What does it have? It has anti-B. For blood type B, it has anti-A. For blood type AB, it does not have anti-A, it does not have anti-B. Otherwise, that would attack your own red blood cells. So the most common, so this is Philippine data, the most common blood type in the Philippines is blood type O, 44 to 46%. Second, or in second place, or interchangeable for second place, third place would be your blood type A, blood type B, and the rarest one is blood type AB. So we know that the blood typing is important because it allows us to determine which blood type will be compatible for blood transfusion. And so this table summarizes that. But I'd like you to keep in mind that when we say blood transfusion, we are referring usually only to the transfusion of RBCs. So when we say that blood is being transfused into a patient, we usually mean that it's the RBCs. We are giving RBCs, pure RBCs, to the patient. So we are not really concerned so much with what's in the plasma of the donor, because that will get washed away in order just to get the RBCs, and only the RBCs will be given to the recipient. And also another caveat is that as much as possible, we give exactly the same blood type to a patient. Although, like in this example, theoretically type AB can receive all of the different blood types. Clinically, we would only want to transfuse type AB, exactly type AB blood into someone who is type AB. So this is to avoid any other potential, even minor reactions. So, but perhaps in an emergency situation, this would be useful. So anyway, let's try to summarize this chart. So of course, each blood type can receive blood from its exact blood type. Type A can receive blood from type A, type B from type B, etc. So let's look at your type A. So type A blood, of course, can be given to type A and also type AB. So type A RBCs can be given to someone with type AB, because type AB does not have anti-A or anti-B. So it can receive type A blood, type B blood, and of course type AB blood. For your type B, of course, it can be given to type B and then to type AB. Type AB RBCs cannot be given to type A, type B, type O. Why? Because type AB RBCs have A and B antigens. Type A has an anti-B antigen, so it will attack the AB RBC. Type B has anti-A, so it will attack the type AB. Type O has anti-A and anti-B antibodies, so it will attack the type AB. So it's not compatible with type A, type B, or type O recipients. Type O, the RBCs do not have either the type A or the type B antigen in its cell membrane. So the antibodies, the anti-A and anti-B antibodies seen in these blood types will not attack the RBC. So it's called the universal donor. While your type AB, because the plasma does not have anti-A or anti-B, it's called the universal recipient. So what happens if there is incompatibility in blood transfusion? What we usually observe is agglutination. So agglutination happens when the antibodies, the anti-A or the anti-B, attacks those RBCs and cause them to clump together. For example, here is your type A blood. Type A blood mixed with anti-B antibodies, you don't see a reaction, but type A mixed with anti-A antibodies, you see the blood cells clumping up. This is what we call agglutination. We observe the same if type B is given to anti-B or is exposed to anti-B antibodies. And this is your type AB red blood cell mixed with anti-B and anti-A. So you observe agglutination in both A or B, anti-A and anti-B. So this agglutination can be dangerous because these clumps of red blood cells can cause plugging of blood vessels, and so it could harm the circulation, might even cause strokes or heart attacks or lack of blood flow to important organs. And the transfused blood will be destroyed by those antibodies and the immune system. And if the kidney is affected, if the agglutination also affects the blood going to the kidney, you can have kidney failure and other organ failures. So another blood type system is your Rh system, and your Rh system is composed of 50 antigens with 6 common ones with the following designations. The capital letter indicates the presence of the antigen, the small letter indicates the absence of the antigen. So of these antigens, it's the D antigen that is more prevalent, more antigenic. That's why it's the more clinically relevant one. So when we say someone is Rh positive or Rh negative, we usually are referring to the presence or absence of the D antigen. So in the population, Rh negative is comprises around 15% of Caucasians and less than 1% in Asians, including Filipinos. So if you have Rh negative blood, make sure to join the Rh negative groups so that you will be in contact with people who are Rh negative, just in case you need blood donation, or maybe you can donate blood to help other people. So one of the feared outcomes of an Rh incompatibility is erythroblastosis fetalis, or hemolytic disease of the newborn. This happens when the father is Rh positive, the mother is Rh negative, and thus they produce a baby who is Rh positive. So while the baby is in the womb of the mother's immune system develops an immune response against the D antigen in the blood cells of the baby. So the mother will be developing antibodies against the D antigen, and so the RBC, the blood cells of the baby, will be attacked, will be destroyed by the immune system of the mother. The incidence rises with each subsequent pregnancy because subsequent pregnancies, so it's like repeated exposures to the antigen, so the immune system develops a stronger and stronger response to that antigen. So the first baby may turn out well or with just few symptoms, but the subsequent babies might be experiencing a stronger immune response from the mother and would present with more severe symptoms or even death. So what happens to the baby? So because the red blood cells are being destroyed, there will be anemia, a large liver and spleen, because remember, these are important in RBC synthesis and also recycling. There will be a lot of nucleated RBCs because the mature RBCs are being destroyed, so there's more production. There's more stimulated production of RBCs, so you now have more immature RBCs. And when you destroy RBCs, remember, you're also producing bilirubin as a byproduct. So if you have too much bilirubin, that may also deposit in the brain and cause damage to the brain. And if the symptoms are severe, or if the immune response is severe, it may even cause intrauterine death. So death while the baby is in the womb. Time for our last concept checkpoint. Pause the playback and try to answer the following questions before proceeding to the next slide. That is the end of our lecture, and these are our references. I based it mainly on Boron and Guyton. Thank you very much for your attention.